Hello, everyone. Good morning, good afternoon, good evening, or wherever you're joining us from. I'm Ara Khachaturian, and on behalf of the Campaign to Prevent Alzheimer's Disease, the Brainwatch Coalition, and the International Neurodegenerative Disorders Research Center, welcome to today's webinar, Empowering Proteomics and Transcriptomics in Alzheimer's Drug Discovery, Insights into the Data Pipeline.
And I'm Ornit Chiba-Falek, joining from Duke University. We are so glad you are here because while data pipeline might not sound like a page turner, trust us, it is the beating heart of Alzheimer research right now. It is where biology meets precision and where biomarker can either unlock the next breakthrough or stall a decade of work.
Exactly. Today's session brings together experts across disciplines to talk through that journey from sample prep and sequencing to data harmonization, analysis, and storage. And yes, we're here to surface the pain points, highlight solutions, and hopefully spark a few "what if we tried this" moments along the way. And this moment couldn't be more pivotal. After years of struggle, the field has celebrated long-awaited progress in terms of Leqembi from Eisai and Biogen, and more recently, Kisunla from Lilly, both earned FDA approval. These are monumental achievements, to be sure, results of decades of work, and we absolutely recognize the significance. But we also know we are not done. These therapies bring promise, but they also raise questions about access, monitoring, safety, and who benefits most. They mark a turning point, not the finish line. The task now is to build on that foundation and to sharpen trial designs, reduce variance, and personalize therapy.
And that is where proteomics and transcriptomics come in. They offer the chance to decode the biological complexity of Alzheimer's at much deeper level to help us understand not just if a drug works, but why, the mechanism, and for whom. That is not just scientific challenge, it is systems challenge. And that is what today is all about.
Exactly. So our conversation today is grounded in the recent editorial Amplifying Efficiency and Accuracy in Dementia Drug Development. This was published in October of 2024 in the Journal of the Prevention of Alzheimer's Disease. A few quick headlines from that piece. Over 95 phase 2 and phase 3 trials have failed in the past decade, costing over $42 billion. Alzheimer's drug development still takes on average 13 years and 5.6 billion per therapy. And yet the biological heterogeneity of AD and ADRD continues to outpace our tools for measuring and managing it in trials. But there is hope. Omics technologies, proteomics, transcriptomics, and beyond are emerging as transformative tools to stratify patients, define therapeutic targets, and reduce trial failures.
Correct. Before we dive into the discussion, let's take a moment to introduce the brilliant group joining us today.
First up, we're thrilled to have Dr. Allan Levey, Professor of Neurology at Emory and Director of the Emory Alzheimer's Disease Research Center. Allan is an internationally recognized leader in neurodegenerative research, and he leads one of the key NIH-supported AMP-AD efforts focused on discovery proteomics.
Joining us from Mayo Clinic, Dr. Minerva Carrasquillo is a geneticist whose work focuses on multi-ethnic longitudinal biomarker for Alzheimer. She is part of the PI team for ClearAD and brings vital insight into how we tackle disease heterogeneity through omics.
Also from Duke University, and correct me if I'm wrong, just down the hall from Ornit, is Dr. Michael Lutz, Professor of Neurology and Pathology and core leader of the Data Management and Statistic Core for the Duke UNC ADRC. Mike has been deep in the trenches of early neurodegenerative disease analysis and knows better than anyone what it takes to make omics data usable.
And rounding out our panel is Dr. Jean-Marie Bouteiller, a research associate professor at the University of Southern California, where he focuses on high-performance computing, model development, and the infrastructure beyond large-scale data analytics. He is also a member of the INDRC Executive Board and brings the computational system perspective we need for today and for our roadmap. So I just want to emphasize before opening today's discussion that today is just the first session in a webinar series discussing proteomics and transcriptomic utilities to advance ADRD drug discovery in personalized medicine ecosystem. So before we go any further, I want to take a moment to thank Jennifer Bird-Derrick and our colleagues at Frontier in Molecular Mechanism of Neurodegeneration for their co-sponsorship at this event and for their partnership in advancing this important conversation. Their support, along with the opportunity to align this webinar with ongoing research topic in helping turn today's discussion into tomorrow's discovery.
Thank you. And I would like to thank the supporters of PAD 2020, the Brainwatch Coalition, INDRC, and members of the Clara Project, as well as everyone attending today. This is intended to be a somewhat of a conversation. Hopefully, it's a lively discussion, and we really look forward to trying to use the platform that Frontiers has put together in a way to have at least the beginning of a conversation, and so that we look forward to hearing your questions towards the end. With that, let's get into our first topic, which is the current landscape of the data pipeline. What's working, what's evolving, and what still needs building?
Thank you, Ara. So to start us off, we are turning to Dr. Allan Levey. Allan, could you set the stage for us? What are you seeing in the field right now, especially when it comes to generating and analyzing proteomic data? And maybe to start the stage, the foundation for this discussion, can you please give us a brief description of TREAT-AD and how genetics, transcriptomic, and proteomics data are impacting target and biomarker advances?
Oh, thank you, Ornit and Ara, and I'm happy to join this panel. I'm really excited today. This has been a topic I've been passionate about for over 10 years. As Ara explained, the failure of amyloid therapies in years past, together with the growing opportunities to have data-driven science inform us about the disease, particularly genetics, as we've begun to learn about the complexity of Alzheimer's disease roots. We have well over 75 risk genes that have been identified, and lots of different biologies appear to be linked to those. And so there's been this clear need to expand the therapeutic landscape and biomarkers beyond amyloid and tau. And out of one of the NIH research summits for Alzheimer's disease over 10 years ago, one of the priorities was recognizing the complexity and heterogeneity of the disease. We needed to start with postmortem human brain. And the Accelerating Medicine Partnership for Alzheimer's disease was formed as really, in my mind, a transformational approach to team science, collaboration, big data. And it rested on the principle that we need to learn more. And before we can really develop clear hypotheses and animal models and the like, we need to understand what's happening in the human brain. And that brings limitations. But what was assembled were a handful of teams from across the United States and many European colleagues. And the teams learned to work together and collaborate. And the teams had not been interacting, but they worked on shared postmortem tissues that were collected from well-phenotyped individuals during life. So I think for me, one of the starting points that's really an important principle as we continue to go on this journey of learning from transcriptomics, proteomics, genetics, lipidomics, metabolomics, all these omics, the starting material is really crucial. Having really well characterized cohorts, having multiple cohorts that can serve as validation, replication, and add to the necessary heterogeneity of tissues collected, ancestries, risk factors for the disease. That became really crucial. And boy, is that a challenge. The AMP-AD was able to assemble several thousand brains from these cohorts, but it's a huge lift to get all the data that really informs the science. And, when I view the omics, at least through my lens, the genetics has been foundational. We know that there's a very large genetic component, but we know that every cell in the body has the same set of genes present in the same genetic variants. And, what we've seen is transcriptomics really become the first hugely powerful platform. And it continues to evolve because we can interrogate genome-wide those genes that are expressed through measuring the transcripts. And we can even do that now in single cells to learn the cellular basis of which genes are getting turned off and on, upregulated, and which ones are linked to these traits from the cohorts. And that's been a very powerful strategy. If you ask any major pharmaceutical company that's interested in developing new drugs, having a genetic anchor to a target is a great way to improve the chances of having a effective, efficacious clinical trial and a drug that works. And that's been tried and true, but it's not perfect. So EQTLs, that is the transcripts that are linked to the genes and those that change expression in disease, finding those EQTLs is a good way to enrich for novel targets. Proteomics is emerging as yet another layer that's really powerful. We've all known that the targets of most drugs are proteins, and the proteins are the real executioners of disease pathophysiology. And we're limited by not being able to go genome-wide, but as we've learned the proteome, that the really powerful new insights into more direct linkages to the complex biology that it can illuminate, and also identify biomarkers and novel therapeutic targets. And I think about these additional layers that are coming, like metabolomics and lipidomics, as also not being able to go as deep as genome-wide, but really powerful integrators of homeostasis and metabolism that are crucial. The real opportunity ahead is now to continue to grow this. I think it's important that we recognize we're still really early on. So we have tools, but I think our understanding of the disease is still quite limited. And that we have a great opportunity to use these new and emerging tools and new analytical techniques that we'll talk about, I'm sure, today, to inform and prioritize the selections going forward. So I'll stop there.
I think this is going to take us also to the landscape of the heterogeneity of Alzheimer's disease. So Alzheimer's disease is not a single disease, it's a multiple disease. And to tackle and fight the disease, we need to recognize that there is no single silver bullet. We've been talking about different diseases, which mean that we're gonna develop different set of biomarkers and different therapeutics and therapeutics targets. So if we're looking at the basic genetics and the correlation between genotype-phenotype correlation that we study in our one-on-one genetics, we know that there is always also correlation between the genetic mutation and the phenotype. And there are mutations within, even in monogenic diseases, even within the same gene, that will extract different molecular mechanism, and the outcome will show us a range of disease severities. So now when we're taking it even one step forward into complex genetics and multifactorial and polygenetics diseases such as Alzheimer's disease and related dementia, we are even increasing further the complexity and the need to look at these diseases as a group of heterogeneous diseases, variety of diseases. And recently there have been few publications from multi-omics dataset, different multi-omics dataset from genomics, transcriptomics, proteomics, even metabolomics and lipidomics, that were able to define subgroups of molecular subgroups of Alzheimer's disease and relate them to the heterogeneity of clinical phenotypes and pathological diversity between the disease. So we know that within Alzheimer's disease, we have co-pathologies, patients with different co-pathologies. So we're talking about a range, and also comorbidities. So there are several diseases, and here with our multi-omics data set, we can link them to different molecular subtypes and tackle our through our roadmap how we're going to fight this disease as a group of diseases. And today is our first session within a long roadmap that's going to take us from from sample collection, basically from the clinic, where we're going to collect different biospecimens, all the way that involving bench-side experimental data set to collect the data, to generate this multi-omics data set, and then followed by computational analysis, more of the dry lab. And then we'll take a circle back to the clinic when we can use our developed biomarker in clinical study and also in early diagnostic, and even to evolve them into novel therapeutics target that will help us to fight the disease on a personal level. So with this disease heterogeneity, I would like to transition to our next topic about data generation, and Dr. Minerva here involved in ClearAD. I would like for you to talk a little bit about, as a startup, about the project of ClearAD, but also give us some highlight about disease heterogeneity and the implication for clinical trial design and how you approach it through ClearAD, and some lessons from multi-omics data project that you were involved in through the years and in bridging the gap.
Thank you so much for this opportunity. As you mentioned, I am one of the co-principal investigators for ClearAD, which is centrally linked longitudinal peripheral biomarkers in multi-ethnic cohorts. Study is an NIH funded U19 program that is, as the title suggests, focused on identifying novel biomarkers that can inform also the development and identification of novel therapies. And this is including several cohorts from multi-ethnic populations, including African Americans, Latin Americans, and non-Hispanic whites. Throughout the program, we're generating multi-omics data, integrating it with clinical data that has been obtained longitudinally for many of these cohorts, such as ADNI and ADRCs. And this includes neuroimaging, cognition, as well as clinical information about comorbidities that we know are going to be important for the risk of disease, and including environmental, social determinants of health that we know are going to influence this heterogeneity that you're referring to. So beyond the complex gene-gene interactions that we know exist for Alzheimer's disease, there are also gene-environment interactions that we're beginning to tackle through this program. The program is composed of 3 cores and 3 projects, and the cores are really mainly focused on supporting the work of the projects. The first one is the admin core, then that coordinates all of the efforts. The omics core is focused on the generation of harmonized multi-omics data, which is a very important topic that I know we're going to be discussing today. And the analytics core is focused on designing pipelines that can analyze all of the data that we're going to be evaluating in a harmonized fashion across all of the projects. Project one is focused on postmortem cohorts, looking at various regions of the brain, so that we can then, in the longitudinal cohorts assessed in project 2 and project 3, we can link back to those signatures that we're seeing in the brain, so that we can really identify peripheral biomarkers in blood that are a reflection of the pathophysiology of the disease. So project 2 is primarily composed of ADNI, longitudinally ascertained study participants who have provided their blood samples for us to generate this set of multi-omics with their exquisite imaging data as well as cognitive data. And then in project 3, we're focusing on the underrepresented populations that have higher risk of Alzheimer's disease, such as African Americans and Latino Americans, who have been ascertained through 5 different ADRCs, including the Mayo ADRC, Indiana ADRC, Michigan ADRC, one Florida ADRC, and the Night ADRC. We are also collaborating with the Emory ADRC so that we can obtain data, multi-omics data that has already been generating for some of their participants. But ultimately, we're going to be integrating data that is genomics, transcriptomics, methylome, proteome, lipidome, metabolome, together with all of the neuroimaging, cognition, comorbidities, and the social determinants of health that have been reported for these participants longitudinally. As you can imagine, this is a huge undertaking, and we are very excited that this is a very strong collaboration between literally 13 different institutions, over 100 investigators that are contributing their expertise to this project.
Thank you. Minerva, if I may ask a quick question. I think you kind of touched on it a little bit, sort of the size of the effort involved. I don't know if everybody truly appreciates what this looks like, and maybe perhaps Allan and Mike, you can kind of jump in on this as we're talking about sort of the various the proteomics platforms. I think, Allan, you had mentioned that earlier, but even going across all the different means of generating data just to collect that, and then the next step, which is then to process it, is phenomenal. Minerva, what is like the scale of this, if you can sort of quantify it for the uninitiated?
Yeah, so we're estimating that just for starters, we're going to have hundreds of terabytes worth of data that we're generating in multi-omics alone. So finding the correct way to store this data in a fashion that is vetted by NIH, and also that is amenable to this collaboration across institutions, and sufficient computational power for all of the very intricate and complex analysis that will be performed. It's something that we are still in an ongoing conversation with NIH, Sage Bionetworks, who's part of this ClearAD program, to help us with data deposition and sharing.
Yeah, the idea of these platform incompatibilities, lack of standardization, challenges and harmonization. What are we looking at?
Yeah.
Especially across different multimodal data sets we're talking about here.
Before we even get to multimodal, I think right now the proteomics platforms are probably the most complex because there are so many different platforms just for proteomics alone. One approach that gives us the greatest depth is the SomaScan, SomaLogic, which uses aptamers. So it's a nucleic acid-based slow off-rate binding to, I think, the latest generation is 11,000 proteins. So it's getting up to about half of the genome wide at that level, which means we've still got a long way to go. But there are 2 other major platforms. One is Olink, which is an immune base. So this is, again, like SomaScan, it's a targeted platform. So these are not really unbiased. They're taking things and developing probes, expecting you know what you're measuring. And so Olink is the immune-based assay. SomaScan is an aptamer. And then there's a third platform, which itself is a suite of different approaches, which are in many ways independent, and that's mass spectrometry. We've all known long that mass spec is the true gold standard for identifying chemicals based on their mass to charge ratio, but it doesn't give you the depth, typically. In tissues, one can get about 10 to 12,000 proteins identified, much like SomaScan. So it's very good in tissues, but as we try to go to biofluids like blood, it becomes a real problem to use mass spec to get terrible depth, great depth like SomaScan. But the platforms are all evolving. And so for mass spec alone, there are several, at least 3 or 4 different sort of mass spec approaches, which are really independent. And every time you read a proteomics paper, you have to consider the different approaches that are being used. We've spent time comparing across these 3 major platforms, and that's where I think we can be most comfortable when we know certain biomarkers replicate across platform and replicate across cohorts. It gives us comfort.
Right.
But the challenge is it's going to continue to evolve, every single day. There's new advances in the technology. The other thing that I think is really important to consider is we can't overlook the importance of sample collection and processing early on. For example, if you look at a study with 10 or 20 sites, you can identify the first set of differences you see at the proteome are distinguishing which sites collected the blood. So more important than which one's there is making sure they're processed in a very similar manner. So lots of traps along the way, but huge opportunities.
Yes, that's a great point, Dr. Levey. I did want to ask, you brought up an excellent point. So if one ran a study on, say, using the SomaScan platform and then was looking for replication studies, what considerations would you keep in mind doing that if you're looking for something to replicate your findings?
Yeah, that's there's a fantastic new consortium which I think is going to be another model, and so it's called the Global Neuroproteomics Consortium, and there have been over 40,000 blood samples now collected from I think 22, 23 cohorts across the world. And really importantly, Michael, it crosses disease. So it's not unique to just Alzheimer's. I think part of our problem in the past is not looking broad enough, particularly when we think about heterogeneity. And so this includes Parkinson's, ALS, includes different sample types. And really the first step, at least our team, so people on our team like Eric Dammer have just done great work developing the tools to do this harmonization across proteome. And I've seen him take those 40 different data sets, and the first thing is trying to regress out all the differences. And immediately you find which sets of samples versus plasma. And again, you find these sites. And so it's an extraordinary amount of work to be able to, harmonize that first layer of analysis.
This is a great transition point to talk a little bit about data analysis. And Jean-Marie, you are sort of on the another sort of node of this effort in terms of looking at these types of data. You had made some comments about RNA sequencing and phenotype data, and thinking about how we can start applying analytical approaches to these data as well. Where are we standing, and what are some of the challenges we're facing?
Yeah, so I just want to, so there are a few keywords that I thought were important that were said earlier. The first one is big data, right? We're dealing with a lot of data, and we have all of these platforms that we hope can learn, we can learn from, right? So we've got these new tools that allow us to retrieve important knowledge about the system we're studying. But then how do we go? I mean, if you have this pry bar, how can you increase the strength that you apply on the system? Well, this kind of leverage you can have if you if you aggregate multiple studies, right? It allows you to look at the system from different angles. And but that can only be done if the data you're working with is harmonized, if you are sure that you're looking at the same, the image that is shown to you from that angle is what you think it is. And I think we've all come across difficulties reading papers. Why? Because there's a matter of interpretation in a paper, right? Something that computers or computational methods are now trying to alleviate by adding objectivity. And So a lot of these harmonization aspects, I believe, need to go through these boxes, making sure that we follow some kind of ontology to make sure that we understand what exactly is being done. And an important aspect, I think we often realize as scientists, is that not only is it important to talk about or to focus on the points we're trying to make, but it's also important to describe the failures or the missteps, things that didn't work, things that were not included in that particular data set, because there may be important aspects there. So I think really having an exhaustive description of what is being done is really useful. And I think to that aspect, there's a tremendous amount of work that was done in systems biology with the systems biology markup language, for example, in which they've created this language that allows you to describe pathways and sets of reactions. And it didn't work. At the beginning, there was a first version, a first dictionary that was used, that was version 1, and it focused on certain aspects. But then, it needed to be extended to include other aspects. And over time, there are iterations over this language that allows us now to make sure we understand. So every model is using a particular version of the SBML language, and it's now very clear if this model is to be used by another group, it's going to be relatively straightforward to implement it. So I really think that this is an important aspect, because we want to take advantage of multiple studies done by multiple groups. And
Right.
also, so big data is and this will be my last comment, is the fact that, so we had the era of big data, now we have machine learning and AI. And AI that's how we can use AI, is through these big data sets. But in order to obtain those big data sets, we need to make sure they are harmonized.
Right.
So that's a really important aspect, especially on the eve of using AI for these things.
Thank you, Jean-Marie, for the insight. Mike, can you tell us a little bit about your experiences from data analysis using multiple data sets that coming from different resources as practically to the research and the pain points that you are experiencing in doing this research?
Sure, thank you. And I do appreciate the opportunity to participate on this panel today. And I second Dr. Levey's point about genetics being foundational for this field. It's been fascinating to see the evolution of research in Alzheimer's from the very early days of ApoE to genome-wide association studies, and now to the multi-omics and single cell area. I think when you consider your first steps in designing data analysis pipelines and statistical analysis plans, you focus on statistical rigor, replication, and finding the best methods that are computationally tractable and scalable. Replication is always critical. And while I think at least the initial forays to find public data are straightforward, finding the appropriate replication data set is going to be a lot more challenging for some of the issues that we've already raised. I kind of call it the Goldilocks problem. You can find data sets that are close, but not quite right. Maybe the assay coverage misses key analytes. Phenotype harmonization is very important and not always straightforward to do. You may be missing key covariates, and that requires contacting authors. So again, building your statistical models requires not just the data, but also a lot of the covariates associated with the data and the clinical data. And that can be a key pain point. On methods, I think the R community is great. It works on a lot of different computational platforms. And often you can find a good starting point, if not exactly what you want. But sometimes the code isn't maintained well, and it may not play well with other code. Plus, there are dependencies on specific R libraries that can be an issue. I think you could say similar things about some of the Python community as well. These are indeed communities where there's a lot of sharing of resources and information, which is helpful. And then once you get plugged into them, this can help a lot. But again, you have to then to match this to your specific problem. I think it's also, and I think we'll probably discuss this in a little more detail, the question around scalability. Was the method tested on a similar scale project? If not, tracking down the problems can be pretty challenging. It's great if the approaches and the software can provide something like a pre-flight tool to test if your data set is formatted correctly and is likely to work. Some imputation software has provided this functionality for genetic data, and I think that's been particularly helpful. So those are a few of the pain points. Good replication data sets, finding scalable methods, debugging and working through those methods, and getting pieces of an analytical pipeline to work together, I think are some of the pain points.
And just to extend on this is emphasize the importance of transparency and a good data management, so the community, the research community, can enjoy data and analytical tools that have been developed in different groups. And if we even scale it geographically and taking it from national effort, and we know that we have effort in the US, we heard about it from Minerva and from Allan, related to AD knowledge. There are also efforts like this in Europe, and I'm sure in other places around the world. So what's your perspective on how we can gather everything together and to make one-stop shop where we're going to have everything, all data set with the method, and that we took from the beginning, from a sample collection, through generating data, through analytical tools, and generating our outcomes, how we can generalize it and make it, globalize it. Any insight from any of you?
Yeah, I'd like to actually tackle that question because I think
Great, Tara.
I think that this is sort of, it comes back to what you said earlier, Ornit, which is this conversation is just that. It's the first, hopefully, of many. And while we're very well represented in life science and basic research on this panel, we definitely need to bring in more of our colleagues from math, from physics, from computational science. We have colleagues in the Czech Republic who are, using some phenomenal methods around areas such as topographical data analysis. And so there are many, many tools that we probably need to bring together. And so, thank you to Frontiers for hosting this meeting, and also perhaps other journals will sort of join in and start to perhaps have more of these webinars where we can have these global papers sort of evaluated with experts and sort of do a real-time peer review almost. And I can just sort of make a statement. I think that, Minerva, you had mentioned the issues around sort of in terms of the data archiving and the storage in the terabytes. Jean-Marie, Mike, Allan, after terabytes, I think, what is it, yottabytes and then petabytes, or I may have that reversed. But in terms of looking at some of these complex interactions, how quickly, what type of time scale are we saying that, perhaps maybe we are now burning past present compute power? Is it in a couple of years, decades, maybe months? I don't know.
We are dealing with a very large data sets, and it is taking it's not something that we can easily play with at this point. I think we can anticipate that both hardware and software will allow us to move faster and allow us to analyze this data, integrate this data. But it is a hard problem to tackle, and at this point, so there are ways to simplify it, right? But it's still something that we have to deal with, and yeah, I don't see any other way. But again, I think we can also take advantage of multiple competencies and all the advances that are taking place in different fields. So machine learning being one of them. Quantum computing is also another aspect that can easily help us analyze this data.
In the single cell world, it is the same sort of situation. Now when we're getting up to sample sizes of several hundred by the cell types, using a lot of the typical Seurat type objects where you try to put everything together in one object, you outstrip most cluster or cloud computing architectures. So you have to get clever about how you break the problem up.
I was going to say, I've seen a growing trend towards databases data platforms being developed so the data don't have to be moved around, where external users can come be hosted, in larger compute facilities, and supports provided. The Alzheimer Data Initiative, ADDI, is one example of that. And I think there are others that are probably emerging. I think it's a great way to drive collaborations. Most investigators can't afford the compute.
Yeah. I think collaboration is the key word. It's not only that it's not a single disease, it's multiple diseases, but we need also multidisciplinary teams and collaboration to bring different expertise in order to tackle all the challenges around the roadmap that we sketches to bring it from the bedside all the way back to the bedside, but with stations around experimental sample collection, a bench-side experimental for data collection, analytical tools for computational, and then the beneficial impact to the patient.
Well, and that's a good place to transition to a question from Barbara Pizzini. She asked the question, what are the ethical considerations and best practices for sharing sensitive patient data related to selectively vulnerable brain regions? Excellent question. Minerva, I see your head nodding up and down. Maybe you want to take a...
Yes, so that is a very important question. And generally speaking, we have to make sure that there is no PHI that is shared. And many data repositories are available that are vetted by NIH that allow you to deposit the essential information that you need from the individuals whose samples you're analyzing, while preserving confidentiality, ensuring that those vulnerable populations, for example, people above the age of 90, we don't report their ages as 91, 92, we say 90 plus. That's just that one example. There are similar issues around, for example, neuroimaging. There are huge efforts now trying to dephase some of these neuroimaging scans to protect the patient's confidentiality. So there are many ways in which we are working together to protect the patient's confidentiality, but ultimately, it is also important to make sure that wherever we deposit this data is also secure. And there's always this database, this data repositories require an agreement, data use agreements from anyone who try to access the data. So there are ways that we ensure that confidentiality of the patient is safe.
Thank you, Minerva. I want to bring another question about integration or basically transferring data sets from post-mortem to in vivo, to live subjects. Allan, do you have any suggestion or any insight?
Yeah, we've been thinking about that question a lot. I'm a a clinical neurologist, and so the motivation for us is we need to bring the, the wealth of insight we get from postmortem brain into living participants. The subtypes you mentioned, for example, are a really key topic right now. We know the subtypes exist at a molecular level from postmortem brain. We've been able to see the same in spinal fluid, so we can begin to do studies of that. But our approach has been to, as I think Minerva mentioned, the brain is really the foundation for, what's a ground truth, for what are the mechanisms of disease that are happening. And our approach has been to integrate the proteome and the other omics in the spinal fluid. It's not typically from the same patient, but do network analyses and understand the pathways that are perturbed and which proteins are conserved in brain and altered in spinal fluid, and then by extension to blood. And boy, it's complex. Some proteins have very direct relationships between brain to spinal fluid to blood, and others are very different. Protein networks in spinal fluid and blood in living patients. Some are very complementary, and some are absolutely, negatively correlated. It's really interesting. And so I think there's a lot of biology, but I think the pathway is there. The blood is really going to be able to be a fantastic window into many of the mechanisms of the brain. I have no doubt about it. And I think, we're learning very quickly the power of just one simple biomarker, plasma p-tau 217, and its great value at predicting whether, amyloid plaques are present in the brain. There are some really valuable data sets that still have to be approached, and there are blood samples from some postmortem tissues available. So blood was collected pre-mortem. And also having blood collected from people that subsequently can do autopsy confirmation is really essential. Again, the heterogeneity that you've referred to is enormous, and we need large sample sizes to really tease that apart. So a lot of work to do.
If I may put in a plug for ClearAD. So project one of ClearAD is actually focused on that, leveraging samples from multi-ethnic study participants that were followed longitudinally in the Mayo Clinic study of aging, who donated their brains. So we have blood collections obtained at various time points, and then the postmortem brain sample, from which all of the multi-omics that I mentioned will be measured and integrated with the neuroimaging, cognitive, etc. The gap right now is that the Mayo Clinic study of aging is primarily composed of non-Hispanic white patients. So what we're doing for that is having in Project One the largest set of brains that we have access to from African American, Latino American donors from the Mayo Clinic Brain Bank, undergoing all of the same types of multi-omics data analysis. And in Project 3, we have the longitudinal data sets for these two populations.
So a long roadmap, but we are already on the road.
A general question, in terms of, we wrote the editorial with an idea that drug development and new therapies are really going to be impacted by the evolution of this technology. What are some strategies that we might think about employing now to help design more cost-effective clinical trials, maybe to maximize post-trial commercialization potential, to provide reasonable evidence data? What should developers and sponsors be thinking about?
I'll be happy to weigh in. Again, it's an area that we've been pretty passionate about. We've had the opportunity to look at the omics from spinal fluid, mostly from a handful of clinical trials, including our own. And I just see enormous potential from enhancing recruitment of the right phenotype of patient to make sure they're in the right window and that they have perturbed pathophysiology that matches the drug target. I think we have good proof of concept that you can enrich and predict responders post hoc. So that needs to be thought about for future prospective clinical trials. Can we start testing some of those things? And I think the other huge opportunity is that drugs have pleiotropic effects. Even if they have a single target, they're affecting pathways. And I think the omics readouts are going to be fantastic ways to actually quantify which pathways are perturbed and restored towards normalcy by different drugs. So again, it's a way to pick out responders, but I think it'll also give a pathway to actually develop better drugs by having an accurate measure for what we should be shooting for.
Yeah,
Yeah,
that's
there's like a context of use, multiple different contexts of use that you've described there in terms of how omics could contribute to clinical trials, both in terms of selecting patients, selecting participants, measuring progression of the disease. It does make you need to rethink greatly, though, kind of your statistical analysis plans and how you power a trial. When you have these considerations involved, because it's beyond just simply random sampling of people and putting them into the trial.
Yeah, so ideally,
Right.
the molecular signatures that we hope to find through ClearID, we can utilize those as ways to stratify patients into the trials.
The patient collection, the selection, I'm sorry, the patient selection is very critical in order to see a signal. Yes, we see it at the beginning of the clinical design, but also the outcomes of the trial.
Here we are at the 56th minute of the call, and it sounds like I hear the outline for another editorial emerging on this very topic, possibly. And so maybe this is sort of a great place to sort of transition over to sort of what our next steps will be, Ornit. What would you like to see happen, and how can we maybe continue this conversation? Certainly, this very last point was fascinating, as were all the comments made today.
Right. So first, thank you all panelists for the great discussion. And we outline here challenges in every step, in every milestone, and entertain some idea how can we resolve it. We would like to continue with this and brainstorm a solution to move the field forward and basically leverage on multi-omics data set on our biospecimen collection in order to push forward the drug development for Alzheimer and related dementia with a development of biomarker based on the multi-omics data set, and then eventually also therapeutics targets. So I think this session is just the first one where we set the ground for the next. And I would like also to see, and this is basically a call for all the audience and of course our panelists to contribute to article collection that we title it Empowering Precision Medicine in Neurodegenerative Diseases via Data Sharing. Data sharing is important, and AI innovation. This article collection will be an initiative from Frontier Molecular Medicine, a molecular mechanism of neurodegeneration section, and it will be co-guest editor by two of our panelists, Jean-Marie and Michael Lutz. So we would like to see a contribution to articles also from the audience that will take us to our next session within this long roadmap toward not only making it possible, but also globalize this effort beyond continent, beyond borders, like to break the walls and to have it as an international effort, one-stop shop. Ara, how you will conclude?
I agree completely. I'm very honored that all of our panelists joined us today. Again, thank you to Frontier. Thank you, Ornit, for your support in this effort, and for the audience. And for also, this will be made available, I believe, also on YouTube at the conclusion. So for those who are watching this after this broadcast, we extend the same offer to you all as well. We look forward to your comments. Thank you.